welcome to what is going to be probably the last revision stream. And what I want to do is I'm going to spend the first 20-30 minutes going over digestion and absorption, and then we'll spend the next 20-30 minutes going over a couple topics which I personally struggle with. So the reason I'm going over digestion is just to keep it in my mind, you know, for the upcoming exam. And then I want to go over genetic variation via meiosis and a couple other things. Because I genuinely struggle with meiosis. I struggle with all the definitions of the words and things like that. It's an annoying topic. But anyways, let's start with digestion. So we're going to start off with digestion of carbohydrates. This is probably the easiest one, alright? Probably the easiest one. So firstly, let's talk about digestion of starch. Which, for those who don't know, starch is then broken down into maltose. And then that maltose is broken down into glucose. And the action of this is due to amylase and maltase. Nice and easy. Starch is hydrolyzed by maltose. Maltose is hydrolyzed by maltase. Nice and easy. The next two you need to know about are genuinely the easiest things imaginable. We could just go, lactose and sucrose. Now, both of these are disaccharides, which means, it's literally just two monosaccharides joined together by condensation. Both of these are nice and easy broken down. Lactose is galactose plus glucose. Sucrose is fructose and glucose. And then, obviously, what they're broken down by, lactose is broken down by lactase. Sucrose broken down by sucrase. And there you go. That's literally digestion of carbohydrates. We can go in a tad more detail. Basically, in the mouth, you have your salivary glands. And these produce, obviously, amylase, which begins the initial digestion of starch. But then we also have pancreatic amylase, which is obviously by its name produced in the pancreas, and that action occurs in the small intestine. In terms of these other two, you can find lactase and sucrase. They are membrane bound. I can't pronounce this. It's disaccharizers. Basically the disaccharide enzyme. Like that. That's how you would spell it. And that's literally the topic. Oh, and obviously in the small intestine. I just put S-int for, for shortening, but yeah. That's literally the whole topic right there for digestion of carbohydrates. We will go back onto this in a tad to talk about co-transport, but that's all you guys gotta know. Very quickly, just some generic keywords, which I want to talk about. This applies for everything. Enzymes, when talking about enzymes and the reaction between it, use these keywords. Obviously refer to the active site, complementary, especially for digestion, ES complex, enzyme substrate complex, which forms when the substrate and the enzyme bind. I guess bind would be another one. If you're talking specifically about why, okay, exam question, hypothetical. Let's say we got a question and it said, why would amylase not digest sucrose? Why would amylase not digest sucrose? What you need to talk about there is how amylase's tertiary structure, its active site, is complementary only to starch, and how because it has its tertiary structure, the enzyme's tertiary structure is only complementary to to starch. I'm trying to think because there was an exam question I did literally the other day which had this exact same question, and I want to make sure I've got the wording right there. Give me a sec, and I'll be able to show you. Here. Right, let me find it. Just a sec. I actually can't show these on streams. Yeah, literally, active site is only complementary to starch due to its tertiary structure. That's 2 marks in an exam, and all you need to put is active site, complementary, and then tertiary structure. So there's your enzyme keyword list. Use that when talking about enzymes, digestion, anything like that. Because genuinely, I'm not joking. When I did that exam there, the one I haven't seen before, I lost 5 marks. 5 marks because I foolishly forgot to use these keywords. I forgot to talk about it being complementary. I forgot to use enzyme substrate complex, and I forgot to refer to the tertiary structure. I could have gotten so many marks, and I didn't. So there you go. There's digestion of carbohydrates. What I might do, I might send this stream to Alex when I'm done, because I know he doesn't really like the digestion topic much. To be honest, I don't particularly enjoy this topic, but it is what it is. It's literally just the enzyme topic, but with a little bit of co-transport thrown in, and a little bit of memorization of one weird process. Protein digestion, this is the easiest one. Guys, this is genuinely the easiest one, okay? This is easy. Alright, ready? Exo, endo, di. That's literally all you need to know. You got your exopeptidases, endopeptidases, dipeptidases. Okay. I forget the order. This is the only thing I forget is the order. Does it go endo then exo? I think it does. I think it goes endo then exo. So what happens is we have here. In fact, I'll draw it in the middle to visualize it better. This is our polypeptide chain. Alright. A very crude drawing. Endopeptidases, if you think about like an endoskeleton, it breaks the bonds in the middle. Basically, in the middle of the... It hydrolyzes the bonds that are in the middle of the chain. Alright? It hydrolyzes bonds in the middle of the chain. Exo is the opposite. If you think about an exoskeleton, like on an insect or something, that would hydrolyze bonds. That would hydrolyze bonds on the outside. So endo hydrolyzes bonds on the inside. Exo hydrolyzes bonds on the outside. Now, you guys will notice that if that bond is broken there, that bond is broken there, you guys will notice that we now have two left over. Two left over. And that is why dipeptidases exist, because they hydrolyze the bonds right in the middle. They hydrolyze the bonds in dipeptides into monopeptides? I think that's a word. Or just amino acids. So there you go. Endo hydrolyze the bonds on the inside. Exo on the outside. Di between dipeptides. One more thing you should note. So remember, this occurs in the small intestine. I think all three of them do, but I might need to very quickly verify that, just so I'm not misinforming you guys. One more thing you might get asked in an exam is something about the stomach. And the thing you need to know about the stomach is it contains stomach acid. So the digestion starts in the stomach. Digestion of proteins, it starts in the stomach. And what you need to know is stomach acid is a very low pH. It's like pH 2 or 3 or something. I don't really recall. Not that it's important. But anyways, stomach acid. That's what you need to know, because obviously if we have like a massive bloody protein, the pH is going to cause things like the ionic bonds, the disulfide bridges to break. And that breaks up a protein into smaller bits in order to increase the surface area for more rapid enzyme action or more rapid hydrolysis. There you go. So stomach acid is actually relevant. Just going to very quickly find out where they're stored. Where it is a small intestine, I'm pretty sure. Yeah, inside / in center of chain, outside in ends of chain. Both of these. Oh, in the protein digestion, you also need to say protein digestion starts in the stomach, where it's partially digested. Protein digestion ends in the ileum. So that's where it's located. That's where all of these things are located. Final thing is lipids. Now this is genuinely where things get difficult. Alright, I'll be real, this is a somewhat difficult section. So let's genuinely break it down into like 3 easy stages. Alright, number 1 is we have our big lipid here. Alright, nothing difficult so far, literally it's just a lipid. Now, here we have our lipase enzymes. I'm just going to draw them as green cubes, okay? Now, obviously, this is a massive lipid. And remember, lipase enzymes hydrolyze the lipids into, they hydrolyze them into their glycerol backbones and fatty acid chains for a triglyceride. Now, look, obviously, this thing right here is big. It's going to take a lot of time to digest it. So that's where we have bile. And I'm pretty sure it's bile salts. So these bile salts are going to go and emulsify the bile salts are going to emulsify the lipid. So now what we have is the lipid is broken up into tiny droplets. That's what an emulsion is. if you want to get a definition for that, it's tiny droplets of one liquid suspended in another. That's what the definition of emulsion is. Remember, the emulsion test is where you add like water and stuff. But anyways, there we go. We've emulsified our lipid into dozens, thousands of tiny droplets. Okay? Good. So now that we've emulsified it, we've increased the surface area. And what this means is it's easier for enzymes to now hydrolyze these individual pieces of lipid. In other words, we have more rapid enzyme action due to the increase of substrates. I don't know how you would word that specifically in an exam. Let me just have a quick look. Emulsification by Boyle. Okay, here. Emulsification. Many small droplets for large surface area. Yeah, for faster hydrolysis by lipase. So there we go. An increased surface area means faster enzyme action. So there we go. I hope you guys are with me so far. So we have a big ball of fat, big lipid. We have to break it down via the bile salts in order for a faster enzyme action. I hope you guys are with me so far, because that's the easiest part. Literally, I mean, okay, hold on. I might have something which could help visualize this, but it'll take a second. I'll go quickly move my camera here. The things I do for these bloody streams. Here. Just a sec. Just a sec, guys. I'll grab it out. Here we go. Which one should I get? I love that one. Okay. Christ, I have not gotten these out in years. Wow. Okay. So. Oh, hold on. If I can actually open it. Okay, I'm gonna have to get a different one because that one ain't working. Come on. There we go. Okay. Sorry about that, guys. I'm trying to do a cool demonstration. I'm trying to be like that, a science teacher over here. Right. What I have here is... For those who don't know what this is sick, right? This is... I used to collect these. This is thinking putty. Right? So this is by this guy who makes like these putties. They're really cool. Back when I was in Florida, I spent about $300 on these. I collected like most of them. So right. This is the example. Imagine this right here is your lipid, right? We emulsify it, and thus we increase the amount of substrate in it, as you can see. If we break it up into tiny like pieces, we are increasing the amount of substrates, and thus enzymes can act on it. I thought I would just show off these, by the way. This is the lava one. Very cool. It's like holographic. I hope that looks good. You know what, I might show off some of these on another stream, but anyways, that's my thinking putty, which I have not gotten out in years, by the way. That's so cool, though. Basically, it's like black on the inside, and on the outside, it's like this holographic red. Look at that, that's sick. Anyways, let's go back to our regularly scheduled revision. See, I feel like I do have, like, ADHD or something. I get so, like, invested in stuff. Right. So, you guys are with me, yeah? You guys understand where all of this nonsense is coming from. Emulsification by bile. That is step number 1. We were to write a 3-step list. First step is emulsification by bile. You guys are with me. You guys are with me. I hope you are, because this only gets more difficult. Step 2. Step number 2. So now, what we have is, we have our glycerol and our fatty acids. They're all broken up now. We have our glycerol and our fatty acid. Okay, they're all broken up. So now, what we want to do really is, why do we digest things? Well, we get these insoluble big molecules and we break them down into smaller soluble molecules. So we've broken it down, but we actually want to use this. Our body actually needs to use this. Our cells need to. So how will we go about that? Well, first of all, we have our bile salts. Did you forget about them? Our bile salts. And what they do is they basically surround our monoglycerides. That's the terminology you should use. They basically surround our monoglycerides. Okay. And what they do is they basically create vesicles containing the monoglycerides. Okay? So the vesicles, that's the next step. Our bile salts form vesicles, and these are called micelles. So micelle formation is the second step. And what happens is the micelles bring the monoglyceride through the cell lining of the epithelial cells. Okay? That's the next phase. It brings it through, and these are obviously going to have your, what do you call it, ileum? Your villi, sorry. They're obviously going to have the villi on. But yeah, what happens is the micelle is going to go through and it's going to release our monoglyceride. And from there, it is reformed by, I believe it's the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum has reformed our triglyceride. It was a monoglyceride, now it's a tri again. And from there, the Golgi apparatus, remember it checks and modifies proteins, it is going to get a protein and it's going to combine it with this lipid, and that's where we get the chyromicron from. And the final step is it's placed in a Golgi vesicle and it's released via exocytosis. One of my favourite words, exocytosis. And there we have it. That's basically it. Step 1. We have our massive lipid. Our massive lipid is emulsified by bile in order to increase its surface area, in order for a faster hydrolysis by enzyme action, by lipase action. From there, we have the micelle formation. Our bile salts will associate with the fatty acids and glycerol, the monoglycerides, to form micelles. From there, the micelles bring through our monoglycerides into the cells lining the epithelial cells lining the ileum. They are reformed, and there we have it. Let me just make sure I haven't missed anything. So lipase is produced in the pancreas, we know that. Lipase is secreted into the ileum, yep. Lipase hydrolyzes the ester bonds in triglyceride. You actually get a mark in an exam for saying that, very nice. What do ester bonds hold together? Obviously your glycerol, your fatty acids. Emulsification by bile. Bile salts are stored in the gallbladder. They are delivered to basically the, the ileum. Bile salts associate with lipids, and they form a vesicle made from the fatty acids and bile salts. They're water soluble, so that's why they can go through the membrane. They are able to deliver the fatty acids / monoglycerides into the lining of the epithelium. And finally, absorption. Absorption happens in the cells lining the ileum. They have foldings to increase surface area to volume ratio for a more rapid absorption. In the middle of the villi, we have a network of capillaries. I'll just draw one. Network of capillaries. In order to obviously decrease the diffusion pathway. Decrease the diffusion pathway and increase the diffusion gradient. Co-transport is required for this. So, very quickly moving on to co-transport, because I hate co-transport, it's a pain. And there may be things which I miss or get wrong. Okay. So, what we have here is this is going to be the lumen of the small intestine. And these are the epithelial cells. So we're going to give them like the villi. Okay. So, what I'm going to do is I'm going to draw our, let's say it's amino acid. That's going to be the amino acids. This is going to be sodium. Right. So first things first, a concentration gradient is established because sodium ions are going to exit the epithelial cell via active transport using a sodium-potassium pump. That's our first step. So a concentration gradient is established because sodium leaves the epithelial cells via active transport using a sodium-potassium pump. Because of this concentration gradient, via facilitated diffusion, I think. Yeah, it should be. Via facilitated diffusion, cells from the lumen of the small intestine are going to diffuse outward using a channel protein. And they are also going to bring with them the amino acids using a symporter, and that's cotransport. But I'm pretty sure I missed out a massive detail there. It's honestly, it's difficult because I don't understand it very well, but I don't actually have anything with me to help. Let me Google. Let me see if AirQA has like a mark scheme or something for this topic. Because what I would hate is to get into an exam and have like a 10-mark question. Well, obviously it won't be 10 marks, but I would hate to have like a 4, 5, or even 6-mark question where it asks you describe how amino acids are absorbed via cotransport, and I have no idea what to say. So let's have a look. So the sodium ions are going to move via a symporter. Yeah, along with the amino acids. And then from there, the amino acids will move via facilitated diffusion out. Sodium will move out via a NAK pump. Let me just have a Google. So, yeah, cotransport. So, active transport of sodium ions from the epithelial cell into the blood lowers the sodium concentration inside of a cell, generating a concentration gradient. We've said that. Sodium ions move into the cell from the ileum by facilitated diffusion. Yeah, we've said that. Carrying amino acid molecules with them via a symporter or a cotransport protein. The glucose concentration is obviously then raised. So yeah, we did it right. We got everything right there, except we didn't specify much. So, if we look, lumen of the small intestine. So now we have our sodium ions in here, which are obviously going to move via the NAK pump again, because there's an increased concentration. Meanwhile, the amino acids, now there's an increased thing of amino acids, so they're going to move via a channel protein. Yeah, so the glucose concentration, the amino acid concentration would increase, and the glucose molecules enter the blood via facilitated diffusion. So literally, it's active transport, created concentration gradients, facilitated diffusion with a symporter, active transport, facilitated diffusion. So it's literally one case of active transport, one case of cotransport, one case of facilitated diffusion. That's literally it. Just make sure in an exam to always refer to the concentration gradient and how sodium ions are used. You get a lot of marks for talking about that. Cool. Alright, I'm happy. Let's move on to some topics I'm not so happy about. The first topic I am not happy with, to be completely honest, is again genetic variation. So what I'm going to do is I'm going to spout out stuff I know, and then I'll spout out stuff I don't know by using Google. So, first and foremost is meiosis. During meiosis 1, that is where we have our crossing over. And what happens is we have we have a pair of chromatids, sister chromatids. And what happens is crossing over occurs where they overlap. One chromosome will overlap to kind of form like this kind of thing. And that forms I think it's like a chiasma. It forms a something. And what happens is then information is swapped between the two that way. And then in meiosis 2, that is where independent segregation happens. So it's where during metaphase they will all line up. I've drawn it with black, so this is going to be impossible. And then what will happen is they will independently segregate so that during the inevitable split, you'll have different ones in each. I've said that in the most vague way possible there, but let me just Google, alright? Let me just Google. So, you have your homologous chromosomes. You have your homologous chromosomes there. They cross over. And that's basically it, apparently. Okay, there. Bivalents develop. Chiasmata forms. Chromosomes break, recombination occurs. So in other words, you literally just need to talk about how bivalents develop, that forms. See, I just don't understand this topic. I think there's too many key words. And the annoying part is, everywhere I look, it gives me different words relating to the topic. Like, some say, it's sister diploid, sister haploid. Others will say bivalent. Others will say homologous chromosomes. Others will say homologous chromatids. I have no idea which one's right, which one's wrong, if they're all right. I'm going to go, actually, onto AQA, because I'm 90% sure there was an exam question last year about it. I'm 99% sure that there is. Let's have a look. Microvilli increases surface area for diffusion. Yep, okay. Anyways. Okay. So, we have our DNA replication. So this is our regular cell. And what's going to happen is the DNA is going to replicate. And then you have your first division. And then your second division. Honestly, the thing I need to know is just chromatid structure, like chromosome structure. Because I always see people drawing it like this, but I know for a fact that's not how it ends up eventually. But I don't know how. Anyways, so yeah, DNA replication, 2 divisions, separation of homologous chromosomes, Separation of sister chromatids produces 4 haploids. See, what does any of that mean? Also, the phylogenetic series, it's grouped according to common ancestry in a hierarchy with no overlaps. Okay, I'm just gonna Google. I'm just gonna Google chromosomes explained. Because I don't understand the chromosome structure. This is what really, really annoys me. The thing I just don't understand, how chromosome structure works in meiosis. How does it work? I don't get it. Okay, so during interphase, we have our chromosomes. See, that diagram does not help at all. Okay, so during meiosis 1, we have our random wiggly chromosomes. They turn into like the X-shaped chromosomes. And then that's what happens. You still have your X-shaped chromosomes. Then in meiosis 2, those X-shaped chromosomes get pulled apart into lines. Okay, I think the best way to explain this now, you know what? Honestly, I still don't understand this. I'm going to be completely... I have no idea what any of this is. I'm going to go on this one website and do a quiz and figure out how little I know. For which term is this the correct definition? A thread-like structure of nucleic acids and protein found in the nucleus chromosome. They're wrapped around histones. A section of DNA that codes for polypeptides or RNA. That's the definition of gene. Each human chromosome is composed of one DNA molecule. No, it's 2, right? Because it's a double helix. The end of a chromosome is, I think, a telomere? Locus. DNA replications during interphase. In eukaryotic, it's linear. In prokaryotic, it's circular and linear, I'm pretty sure. No, it's circular. Prokaryotic have the same bases here. Prokaryotic DNA, no. The names of the extra molecules of DNA, plasmids. The name of a pair of chromosomes which have the same genes in the same loci. So that's homologous. So homologous is a pair of chromosomes with the same genes in the same loci. Overall DNA molecules have an overall negative charge. No idea. You inherit your mitochondrial DNA from birth. Pretty sure mitochondrial DNA is circular. Chloroplast contains circular DNA. Okay. So I probably did alright in that quiz. Despite barely knowing a thing. Yeah, okay. I'm just gonna find a quiz of meiosis. Okay. Meiosis results in its full diploid daughter cells. The picture depicts what phase? It's where the spindles are moving to the equator, and you have four. So that would be prophase 1, right? Because you have 4 chromosomes. Crossing over occurs during. That's prophase 2, right? No, hold on. Metaphase 1. Meiosis produces zygotes. No, sorry, gametes. Oh, okay. So during prophase in meiosis, homologous chromosomes pair up. A cell with a diploid number of 24 undergoes meiosis. So that'll be 12, right? Because if that's the exact same, then that should be 12. The picture depicts what stage of meiosis. So it's 2 cells splitting. So that would be metaphase 2. And I got everything wrong. No, I got those 2, right? Got that right. Got that right. Got that right. I got 1 wrong, but it's saying I got 63%. Prophase 1. I got that right. Crossing over is during prophase 1. Okay, I didn't know that. I got everything else right though. So yeah, if there were let's think it like this. If there were 12 here, there would be 12 there, and then 6 in each one of those. I just need to know chromosome position. If I can do that, I win. I understand it. I will literally be able to understand this whole topic if I just know what the chromosomes look like. If I just know what they look like, I will understand. But there's no diagrams which I'm understanding. I don't understand any of these diagrams. Yeah, I'm lost. Honestly, the only thing I don't understand about this topic is chromosome like position. Because here it's showing how there are, during prophase 2, each cell has the paired chromosomes like so. Okay, hold on. Hold on, I think I've got it. Hold on, I think I've got it. Okay, so if there were, like, let's say 4 chromosomes here, right? Then that would lead to there being, I'm just going to draw it with the exact same. And that means that there would be, oh, and then it's, oh, because, you know what I misinterpreted, guys? It's the fact, I think this has just clicked for me. I think it's the fact that the chromosomes divide here during interphase. Right? So if we had 12 at the start of interphase, this would obviously go back to 24. Okay, so if we had 12 chromosomes right at the start of interphase, they would obviously all divide to get you to 24. Then they split off, they divide, giving you 12 again. But this time, it doesn't divide. The DNA does not replicate a second time. And as a result, that's why you only have this single-stranded. During prophase 2, the chromosomes condense. The chromosome begins moving forward. During metaphase 2, the centromeres of the paired chromatids. So yeah, you'd still have a chromatids, obviously. I'm just going to quickly go on the teacher's PowerPoint, see if I can understand this any better before I stop for the day. Because again, I barely understand this topic. I still don't understand it very well, but... Let me just see. Okay, I'm pretty sure this is a PowerPoint from, like, year 9. Let me just see if... The cell makes an identical copy of all chromosomes. Yeah. Okay, this might be a good diagram. So, the pairs of chromosomes line up along the center of the cell, DNA replication, arms of the chromosomes pulled apart, pairs are pulled apart. Well, hold on, but if the DNA... Hold on. First you have the single-stranded single-stranded things, and then they replicate. So you have the double-stranded, and then they're still double-stranded. I think I need to add a tiny bit to the diagram to fully gauge this, right? So to begin, they're single-stranded. Right? 1, 2, 3, 4. And then they replicate. There we go. You know what? This is clicked now. So they're single-stranded. So I'm just going to draw like single-stranded, like that. Then they're double-stranded, like that. Then they're still double-stranded. And then they're single-stranded. Alright, I've understood this. I'm heading off now.